Good morning, everybody. Woo! Happy Friday. Welcome to another episode of Ion Physiology. Today we're celebrating the end of your first week. Woo, woo, everybody. Today we just wanted to take the time to go through some of the content that theoretically should be worked on during the first week of the program and any topics that we felt were challenging or just are more important and thought that you all would benefit from hearing a little explicitly from us regarding the topics. So we've created a little list. We're going to run through some of them. First up, Haley's going to talk to us about reflection coefficients.
Yeah, let's get into it. So, I'm sure you guys have read about reflection coefficients or sigma by now, which is a value that expresses the amount of a solute that is able to cross a membrane. Now, we wanted to talk about this because it can be a little counterintuitive because with a reflection coefficient of 1, that means that 100% of a solute is being reflected, not that like 1 is getting through or something. It's 100% is being reflected, so nothing is getting through. But on the flip side, a reflection coefficient of 0 means that 0 of it is being reflected, so that all of it is passing through. Um, so, if you were to hear a question maybe sometime in the future about what would be a likely reflection coefficient of carbon dioxide, what would you think that would be? Well, we know that gases are freely permeable across the membrane, so that means it's all going to pass. And you need to immediately think reflection coefficient of 0 because nothing is being reflected. So just to clear the air on that concept a little bit, I think I think that covers it pretty well.
Yeah, thank you, Haley. Thank you.
Thank you, Don.
So, brief aside, we forgot to mention we're missing Dr. Brainerd today. He is at home ill, come down with, freshman fever, first week fever, whatever you want to call it. So it's just us GTAs hanging out today talking to you all. Next, Nadim's going to talk to us about some concentrations. Nadim?
I wanted to touch up on these because they're gonna keep showing up, and that is the extracellular and intracellular concentration of specific ions. There's lots of them, to be honest. There's one, two, three, four, five, six, seven, eight. The most important two I think you should remember are the for the membrane AP are the sodium and potassium concentration. I know we keep saying that this is not about like memorizing whatever, but for this specific subject, I think you should memorize like the concentration of all of these. The thing with these concentration is that they're gonna keep showing up throughout the entire course, and that's why we emphasize that you actually memorize them. Um, Sodium, potassium right now, and then during you know cardiac, you will see a lot of calcium showing up, and you know and then renal, you'll have like a whole lot about HCO3 like bicarbonate, and also the body when it wants to like equivalent like come back to like yeah, like the steady membrane potential, it actually will open certain chloride channels. And so it really is important to memorize where these ions and how to like get it out of the way right now, memorize all of them and keep coming back to them because they will come back at like they'll come back in each lesson. Very important to memorize them. It'll save you so much time later on.
Yeah.
That's about that really.
Yeah, just highlighting sodium, chloride, bicarb, calcium, and potassium. Those are your main five. Those are the ones, like Nadim said, are going to be popping up throughout this whole program. So there's some things you unfortunately do have to memorize, but I think it's just important to remember sodium and chloride always typically go together. They'll be higher in together, higher or lower out together, or I may have said that backwards, vice versa. But
You're good. You're good.
but but yeah, it's unfortunately something you have to memorize. Um, Next, I wanted to talk about these diffusion graphs that we've talked about. We have like simple diffusion, facilitated diffusion, carrier mediated. So one thing that it can be a little confusing is the concept of a transport maximum. So when things have simple diffusion where it's passive, facilitated diffusion where it can be passive. Um, if it's passive, it's not dependent upon anything other than its permeability or via simple diffusion or channel. It's strictly linear. There's no transport maximum. As much can cross depending on the concentration. Whereas something that's carrier mediated, it depends on some factor for it to be able to pass. And there's only so much of like a carrier protein available for something to pass. So if you have all of the carriers used up, like that's the maximum number that can pass. So when you see these graphs, you'll see a plateau, and that plateau is the transport maximum to where nothing else can pass through the membrane because everything's saturated. And now I think Amy's gonna bring us into a little history of physiology lesson.
Okay, perfect. So for the history physiology lesson, this is just from physiology in the previous years this week. So on August 28th, 1952, there were two scientists, Alan Hodgkin and Andrew Huxley, and they published one of the most important papers in neuroscience. So I'm not sure that a lot of you have hit neuroscience yet, but it should be coming shortly if you haven't. So they studied how neurons send electrical signals using a giant nerve from a squid. Nerves from squids are so big, so it was just easier to experiment on. So they discovered that tiny channels in the neuron's membrane called voltage-gated ion channels open and close in response to changes in voltage. These channels then let sodium and potassium ions move in and out of the cell, creating the electrical signal we call an action potential. So this work actually got them the Nobel Prize in 1963.
Very nice. Amy, you're in neuroscience, weren't you? Or am I misremembering?
No, that was
No?
Michelle though.
It was Michelle. Okay. See, the brain was connected a little bit. Yeah, do you have anything you want to add to the history physiology? No. Okay. Well, getting back into first week shenanigans, I wanted to talk about osmosis. And osmosis, it can be a little bit trickily defined. Um, a lot of places sort of simplify it as a like kind of simple diffusion, and that can be a little misleading because water is not necessarily a particle with a gradient diffusing along something. Water just moves, simply put, from where it is to where it isn't. So it's similar to simple diffusion, but that's the most plain way that you can state what exactly osmosis is. Amy, I think she's going to talk to us about active transport.
Yes. So I'm mainly going to talk about the difference between primary and secondary active transport because these can be a little bit confusing, but they're two-way cells can move against their concentration gradients. So first we'll talk about primary active transport, which directly uses energy from ATP to pump molecules across the membrane. A classic example of this is the sodium potassium ATPase pump, which moves 3 sodium ions out of the cell and 2 potassium ions in, and this uses 1 ATP molecule. And in contrast, secondary active transport, it doesn't use ATP directly. Instead, it relies on the gradient that was created by that primary active transport. An example of this is the sodium and glucose symporter, which moves sodium down its concentration gradient into the cell, and along with it, it drags that glucose against its gradient. So just to summarize, primary active transport does directly use ATP, and then secondary active transport uses ion gradients created by that primary active transport.
Nice. Thank you, Amy, bringing in the renal physiology there. That just goes to show you all, like when we say the things that you're learning now are going to keep coming up throughout the entire program, that's just another evidence of it. The sodium potassium ATPase is not going anywhere. It's going to come up over and over and over again everywhere. So yeah, speaking of things that are going to show up again later, I wanted to bring up a feature of cell membranes, gap junctions. So in the lessons, you learned about 4 different characteristics of cell membranes, and gap junctions is one where there's a connection between 2 cells, and this connection allows charge to pass from one cell to the other. So this is via connexons in the gap junction, allowing charge to pass from one to the other. So this is going to come up again in action potentials, cardio, just over and over, neuro, just over and over again. So just memorizing gap junction, flow of charge. And then similarly, I wanted to bring up the actin and myosin framework within cells. So the actin filaments, this is like the cytoskeleton you hear about in the cells themselves, the framework for how everything moves. So in cells, we have vesicles, which contain different things. You will get into that more in exponentials and neuro, but we just want to establish, you have to visualize that actin cytoskeleton, and there's myosin interacting with actin, and they're able to interact and move these vesicles within the cell. So just, it's like kind of like the highway interstate system, that actin and myosin highway system in the cell that's moving things around inside the cell to the cell membrane, away from the cell membrane. We'll get further into that in more detail within the next few sections of the course, but just wanted to emphasize that now. And then I think Hunter's going to talk to us about G proteins.
Yeah, so just going to get into a little bit about G protein coupled receptors. I think, at least for me, this is one of the more confusing things in this unit. So they are a huge family of receptors, and they, in short, basically work by taking an outside signal from the cell and then converting that into an internal response. And the way they do this is through G proteins and second messengers. So when something binds to the outside portion of the G protein, it's going to then activate the G protein itself through some of its various subunits, which can determine the kind of effect that it has on the cell. And then that's going to then cause downstream effects, something like, I think we've talked about like phospholipase C, adenylyl cyclase, stuff like that. And so then that's going to activate second messengers like cyclic AMP, which then can either amplify the signal or cause further cellular responses, like increasing calcium concentrations.
Yeah, that's what
If I can add something, I'm sorry about
go
that.
ahead,
If I can add something about these G-coupled receptors and the other channels open up and allow things in, if you want to think of it this way. What the G-coupled receptors is that they bind to a ligand, they bind to something, okay? And that thing does not actually penetrate the cell, okay? So it'll bind here, and then feel it here, that G-coupled receptors, and then something here will move. So that, like the calcium, whatever is going to bind here. What is the other one? ACH is still calling sometimes.
Mm-hmm.
Norepinephrine as well.
Mm-hmm.
when they bind here, they don't penetrate the cell. Like nothing opens and no, something actually changes here. Like something a signal gets sent within the G coupled receptors itself, and then something changes here inside the cell.
Yeah, very, very important distinction to make, Nadim. Thank you. Yeah, that's what I just highlighting. Like we've talked about channels and aquaporins and things like Nadim said, but this is something totally different, in which a ligand, which is just anything that binds to a receptor, like Nadim brought up acetylcholine, norepinephrine, something will bind to the G-coupled protein receptor, which spans the membrane 7 times and then causes some sort of change that releases those. It's called a heterotrimeric. So there's those 3 subunits that will dissociate. So ligand binds extracellularly, like Nadim said, and then intracellularly, that heterotrimeric protein dissociates on the inside of the cell. And those subunits are what go and activate other proteins inside the cell to have some sort of intracellular effect, like Hunter talked about. So yeah, thank you guys. I feel like those were the toughest concepts, or the concepts that I feel will be coming out more and more throughout the program itself. And I want to thank you all for bringing that to attention. Just closing out, I know it's just our first week, but we wanted to remind you all of deadlines within the program. So there's only one deadline in this first course, and it is October 8th. So everything has to be done by October 8th. You can do it on your own time. You can get it all, you can rush through it all, get it done in 3 weeks. Don't recommend that. You could wait till the end and do it all in 2 weeks. Don't recommend that either. We've made that calendar, that proposed calendar. We just recommend that little by little you work through the content and sit with it and try to get things done. So that way you're not overloading yourself with content or work at any point throughout it. But yeah, so the only real deadline is October 8th. But we recommend that, you spread around the workload, so that way you're not overworking yourself or tiring yourself out, because yeah, it's a disservice to you and your learning to try and cram it all in at the last minute. Do y'all have anything you'd like to add about first week shenanigans?
Everybody keep up the good work. I see y'all moving through it, and we're proud and we're here to help.
For sure. All right, well, thank you guys. It's been a lovely episode of Ion Physiology. Hopefully we'll have Dr. Brainerd back here with us next week, and we'll see you later.